Okay, cellular transport, we're going to be focusing on passive and active transport. So I've got a video here from the Amoeba Sisters. So if you'll take a few minutes and watch that, please. So what we're going to be looking at is we're going to be looking at the movement of molecules across the cell membrane. And when you're talking about the movement of molecules across the cell membrane, you have to talk about concentration gradient. So concentration gradient is the difference in the amount of dissolved substance from one side of the cell to the other. So with this one, we're just looking at little purple circles, but what we see is on this side, there is a greater, or there was a greater concentration of little purple circles, and on this side, there was a lesser concentration of little purple circles. And what seems natural is to move those particles from where there is a whole lot to where there's a few until you can balance them out, which would be called reaching equilibrium. And the same thing on this other side with another shade of purple circles, moving from an area of high concentration to low concentration. And in both of these cases, this is called passive transport because the movement is going from high to low concentration until equilibrium or homeostasis or a state of balance is reached. Now, in this scenario here, is just simple diffusion, in which you see these molecules being able to move directly across the phospholipid bilayer. In the other scenario, we see these molecules needing a little bit of help to move to the other side, and the facilitator or the helper is a protein. So what we're going to do is we're going to look at each of these three types of passive transport. And I just want to make sure that you note that passive is high to low. That's what makes it passive. So we will look at simple diffusion. And simple diffusion is of small molecules that are nonpolar, so they're not going to be reactive with water, and they can go directly across the cell membrane. Um, an example of diffusion is osmosis, and osmosis is the diffusion of a particular or a specific type of molecule. It's just the diffusion of water. And then over here, we see this big, gigantic uh, channel protein, this transport protein, and it is for moving substances that are maybe larger, so larger molecules, or molecules that are polar, so they're reactive. So it has to have this tunnel to be able to get to the other side. But all of these situations here, you're moving from high to low concentration. So on your note sheet, you're going to be building your information about passive transport. So if we're talking about passive transport, we're talking about the movement of these molecules from a high to low concentration. Now that is sometimes referred to a moving with the concentration gradient, going with the gradient, which is kind of like going with the flow. It seems to be easier. It seems a natural way to make that happen. And in both of these, no matter how you're thinking about it, high to low, going with the flow, this is not going to be costly to the cell. It's not going to require energy. And when we're talking about energy requirements, we're talking about ATP, adenosine triphosphate. So this isn't costly to our cells. And the examples again, the simple diffusion. So those are smaller molecules, they're nonpolar, they're non-reactive, and they can slide straight across the cell membrane. Facilitated diffusion needs the help of that protein to help the molecule move across the other side because maybe it's larger or maybe it's polar. And then osmosis, the movement of water. Now, I do have a video here, and please feel free to look at this video. It's called Skittle Diffusion. And then I just have a couple of pictures here. So if we took a beaker of water and we just squirted some food dye in there, some food coloring, we would see that this food coloring is going to move from a concentration or area of high concentration to an area of low concentration until it reaches equilibrium. So that could take a little while. Now for active transport, it's just the opposite. So with these little purple squares here, they're moving from a lower concentration into an area of higher concentration, and they may also require the aid of a protein or a protein channel. And if you also notice, they're also going to need ATP, so they're going to need to use some energy to make that happen. So here we see the difference. One of the big differences is going from low concentration to high concentration and the requirement of energy. And two basic types are referred to as endocytosis and exocytosis. So we're putting our notes together with active transport. We are going from low to high, so that's going against the concentration gradient. It's kind of like swimming upstream. And in both of those cases, when you think about it, my little guy kind of running up this mountain here or swimming upstream, both of those are going to use energy. They're going to cost the cell some ATP, so it is costly to our cells. And examples, endocytosis, exocytosis, and something called the sodium-potassium pump. So if you look at endocytosis, so coming into the cell, we could see these, what do these little blue dots represent? Coming inside of the cell, the formation of little vesicles, and then the same, well, the opposite with exocytosis, these vesicles fusing with the cell membrane and releasing particles. Either way, both of those moving into or out of the cell are going to be costly. And something I wanted to throw out here, the sodium potassium pump. Now, the sodium potassium pump, we're talking about the movement of sodium from one side of the cell to the other. And what you're seeing there, and it kind of happened before I got to say anything, is the ATP molecule broken down into an ADP molecule to use that energy to move those sodiums over to the other side.